Hello, I'll be talking about the complement cascade, or aka the complement system. The complement system is a part of the immune system that enhances or complements the ability of antibodies and phagocytic cells to clear pathogens that may enter our body. It contains both components of adaptive as well as the innate immune system. So let's just start. Okay, I'm going to talk about the complement system, but the important thing about the complement system is that there are three ways of activating the complement system. There is the classical pathway, the mannose-lectin pathway, and the alternative pathway. So these three, 1, 2, 3. I'll talk about the classical pathway first because it was found first historically. So this is how it starts. The classical pathway starts once there is the antigen as well as the antibody complex on the surface of the pathogen, or it could be anywhere. Basically, if you have the antibody and the antigen, the C1QRS complex can form and attach onto that complex. And basically this complex of Q1QRS will convert the C4 into C4A and C4B. Okay, so I'm going to write down like this as if it's floating around in the basically plasma, but actually what happens is that this C4 attaches itself to this complex and it's going to attract other things onto this. So C4B, this is actually attached to the C1QRS nearby the pathogen, and then it comes C2 and becomes C4B2. So it basically makes a complex together with the C4 and the C2. Okay, C4 cleaves away by itself, and the C2B goes away as well. So finally, it makes a compound called C4B2A. It's slightly confusing, but just think of it as the a part of the C4 as well as the part of C2 makes what is called aka a C3 convertase. It's called a C3 convertase because exactly what it does is to convert this C3 into C3b as well as C3a. C3a is pretty important in the way that it cleaves itself and goes away and does a lot of things, like it will excite some mast cells to create histamine. It will go and make blood cells bigger, and it can also signal the neutrophils to come and gather itself near the pathogen, and can also make the capillaries kind of leaky so that the neutrophils can go into the plasma and basically attack the pathogen. Okay, C3b, this is very important because C3b will attach itself to the C3 convertase to become C4b to A 3b, and this is actually called C5 convertase. C5 convertase is also very, very important because what it does is it converts C5 and it creates a C5b. And the importance of C5b is to make the membrane attack complex. Once C5b is around, it will attract C6, C7, C8, and C9 to make this kind of complex. And this is C5b to 9 basically, and these are all parts of C9, which will just sort of like slot in itself, and it creates a pore like that onto the pathogen. Let's say it's a bacteria, it could be a bacterial cell. So what it does is that it would lyse the cell basically by creating a hole, boom, into the cell. Okay, so this is one of the outcomes of the complement system. You can say the formation of MAC. So this cell is suffering from the consequences of having complement system. Okay, so we've basically discussed two outcomes of complement system, which is one is all these chemotaxis, attraction of neutrophils and all that, as well as the MAC. What C3b does as well is it attaches to the cells or the pathogen and it basically makes it visible for like a macrophage to come and eat it up. So that's called opsonization. All right. So far I've explained everything from the classical pathway, but let me explain about the other two pathways of activation. We have the mannose-lectin pathway. You might ask me, what is mannose and what is lectin? Mannose is actually a polysaccharide that is present on the surface of the bacterium, and lectin is basically what can start off this complement cascade. So instead of going to C4 and C2, it's going to be signaling through a molecule called MASB1 and MASB2, which respectively will cause this cascade of events to make the C3 convertase once again. So in this case, in the case of lectin mannose pathway, the C3 convertase is the same as the classical pathway, which is C4B2A. Okay? The alternative pathway, on the other hand, is slightly different. In the alternative pathway, you don't need an antigen-antibody complex. What it needs, however, is circulating C3, which is combined with H2O water, and also needs factor B and factor D. Usually, without the presence of pathogens, this will just disintegrate by itself and nothing will happen. However, if it finds like a bacteria, let's say these are bacteria, and C3 goes, oh, there's some bacteria. What happens is that it will combine with factor B and factor D to become C3H2OBB. And what this does is convert C3 into C3b. And again, with the action of factor B and factor D, it becomes C3BBB. Okay. In the case of the alternative pathway, this becomes the C3 convertase. And so it starts the cascade of events that would form the membrane attack complex. Okay? So basically, no matter how it starts, the alternative, the mannose-lectin, or the classical pathway, it all ends up with three outcomes, which is all this inflammation, chemotaxis, as well as the formation of the MAC, the membrane attack complex, as well as the opsonization. So just to summarize everything, main point 1, 3 ways of activation. The classical pathway, just to remember that the classical pathway needs an antibody and antigen and antibody complex. And it starts with the complement 1 QRS complex. Okay. The mannose-lectin, it doesn't really need antibodies to happen, same as the alternative pathway. Okay, main point 2 is that the 3 outcomes of complement cascade, which is the formation of MAC, opsonization of pathogens, and inflammation. There's one more thing that I would like to discuss, which is the regulation of complement system. How it needs to be regulated so that the complement system don't go around damaging our normal cells. So what it does is that one example is that once there is a CD59 surface molecule on the cell, let's say this is a normal cell, our bodily cell, it looks really innocent. Okay, what it does is that once CD59 is around, even if the C3b to 8 is on the cell, the C9s can't form the pore on the cell surface, so that the cell doesn't lyse or be damaged because of the MAC. Some viruses are very smart in a sense that they will incorporate CD59 into their own cell, the structure, like HIV and CNV. And once this thing is malfunctional, we have diseases such as the paroxysmal nocturnal hemoglobinuria. And basically what happens is that because of the lack of CD59, we have the membrane attack complex on our own cells. And that's why we have some symptoms and signs of PNH. Okay, I think that's all for now. Bye-bye.